Welcome to episode 25 of Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we're starting an exciting journey into the muscular system. And if I'm being honest, this is where this class really starts to get fun for me. This episode will cover the general functions of muscle tissue, the detailed anatomy of a sarcomere, and the structure of the neuromuscular junction. In our next episode, we'll take a closer look at excitation, contraction, coupling. But for now, let's set the stage for that by exploring the foundation of how muscles function and interact with the nervous system. So muscle tissue found throughout the body has three main types: skeletal, cardiac, and smooth. Despite their differences, all muscle types share three primary functions. Force production: muscles generate force to create movement or maintain posture. Movement: skeletal muscles allow voluntary movements. Cardiac muscle pumps blood. And smooth muscle regulates internal environments such as food through the digestive tract. Stabilization and regulation: muscles maintain body posture and regulate openings and closings in the body such as sphincters. Now, most of the remaining part of this episode is going to focus on skeletal muscle. So let's start off by talking about the anatomy of skeletal muscle fibers. Skeletal muscle fibers are highly specialized for contraction and consist of several components. There's the sarcolemma, which is the plasma membrane of a muscle cell, and this is the part of the muscle cell that conducts action potentials. Then there's the sarcoplasm. The sarcoplasm is simply the cytoplasm of a muscle cell, which contains the organelles, glycogen, which is the storage form of glucose, and myoglobin, which is the muscle cell's version of hemoglobin that's found in the blood. Then there are the T-tubules, or transverse tubules. T-tubules are invaginations of the sarcolemma that carry action potentials deep into the muscle fiber. Just imagine them as a hole in your lawn. Okay. Finally, we have the sarcoplasmic reticulum, often abbreviated as SR. The sarcoplasmic reticulum is a specialized form of smooth endoplasmic reticulum that stores and regulates calcium ions, which are essential for muscle contraction. So this leads us to the sarcomere, which is the functional and structural and functional unit of skeletal muscle. Sarcomeres run from one Z disc to the next, and its structure includes several key components. So you have the Z disc, which I just mentioned. The Z discs define the boundaries of the sarcomere and anchors thin filaments referred to as actin and elastic filaments, which are called titin. In the middle of the sarcomere, you have the M line. Okay, the M line anchors thick filaments known as myosin. From there, we've got two bands and a zone. There's the A band. The A band contains the entire length of the thick filaments, or of all of the myosin filaments, and this includes overlapping regions with thin or actin filaments. Then there's the I band. The I band only contains those thin actin filaments and spans adjacent sarcomeres. Finally, you have the H zone. The H zone is actually a subsection of the A band. Now, in the H zone, this is the part of the A band that only contains those thick myosin filaments, and this zone shortens during contraction. Now, the proteins within the sarcomere can be broken up into three categories, and each of these categories has a pair of proteins. First, you have the contractile proteins. So you have myosin and actin. So as I mentioned a moment ago, myosin are thick filaments that bind to the end line, and ultimately they can bind to actin and generate movement. Then you've got actin. These thin filaments serve as the binding sites for myosin heads. When we talk about actin and myosin, I like thinking about myosin as hands and actin as a rope. And so the myosin hands pull the actin rope to cause muscle contraction. But we'll get into that more later. Then there are the pair of regulatory proteins, troponin and tropomyosin. Now, troponin will move tropomyosin out of the way to allow actin and myosin to bind to each other. Tropomyosin, its job is to block myosin binding sites on actin in a relaxed muscle. And then finally, we have the structural proteins. There's titin and nebulin. Titin is an elastic fiber that ultimately helps stabilize those thick myosin filaments. Nebulin, on the other hand, helps stabilize those thin actin filaments and helps align them within the sarcomere. Now, as I mentioned a moment ago, when a muscle contracts, some of these bands and zones change. And so when a muscle contracts, what's happening is that there is a sliding filament mechanism that ultimately causes the sarcomere to shorten. And so here's why those specific bands and zones either change or remain constant. Now, if you remember, the A band runs the entire length of those thick myosin filaments. Now, the A band remains constant during muscle contraction because it represents that entire length. And during muscle contraction, the length of these filaments, both thick and thin, do not change. So as a result, the A band remains constant. The I band, on the other hand, shortens as the thin actin filaments slide past the thick myosin filaments and produces more overlap. As there's more overlap, there's less area where there's only actin, so as a result, the I band gets shorter. Then we have the H zone. Remember, the H zone is that subsection of the A band that only contains myosin. During muscle contraction, the H zone decreases in size or could potentially disappear entirely because the thin filaments are pulled closer to the M line, which increases the amount of overlap with myosin filaments. Ultimately, muscle contraction causes the Z discs to be pulled closer together. This shortens the sarcomere and ultimately the muscle fiber. This coordinated movement ensures that muscle contraction is efficient and forceful. Now from there, we're going to kind of back out a little bit and move to the neuromuscular junction. And this is where the nervous system communicates with skeletal muscle to initiate contraction. So let's explore this area. When an action potential travels down the axon of a motor neuron, it reaches the axon terminal. And from here, for the next several steps, this is very similar to what we discussed when we were talking about a chemical synapse. So when an action potential reaches the axon terminal, voltage-gated calcium channels open. The influx of calcium ions into the axon terminal triggers synaptic vesicles to release the acetylcholine into the synaptic cleft, which is that gap between the neuron and the muscle cell, and this occurs via exocytosis. Acetylcholine molecules diffuse across the cleft and bind to ligand-gated sodium channels on what we call the motor end plate, which is a specialized region of the sarcolemma. This binding causes sodium channels to open, which allows sodium ions to enter the muscle fiber. Ultimately, this depolarizes the sarcolemma. Now, there are several additional steps that occur in the muscle cell, but we'll discuss what happens next in the episode on excitation, contraction, coupling. So let's wrap up with our key takeaways, and we've got 5 of them for this video, or for this podcast. So first, muscle tissue performs essential functions like force production, movement, and stabilization. Skeletal muscle fibers are highly specialized, containing structures like the sarcolemma, T-tubules, and sarcoplasmic reticulum. The sarcomere is the functional unit of muscle consisting of contractile, regulatory, and structural proteins. During contraction, sarcomere bands and zones change as actin and myosin filaments slide past each other, which shortens the I band and H zone due to the increased overlap, while maintaining the length of the A band. At the neuromuscular junction, acetylcholine released by motor neurons binds to receptors on the motor end plate of the muscle, ultimately initiating an action potential in the muscle fiber. And so that's it for episode 25 of Pulse of Physiology. I hope this episode has provided a solid foundation for understanding the basic functions and anatomy of skeletal muscle fibers. Join me next time for episode 26, where we'll continue our discussion that we started about the neuromuscular junction and go into and discuss excitation, contraction, coupling. Until then, keep questioning, keep exploring, and stay curious.